Hello everyone, thank you for attending today's webinar. So I'm Sophia Muret, Project Manager of Sales and Marketing at Aussie Biosciences. And today we are with Cédric Sapé, a doctor in cellular biology and our project specialist. So we are going to start with a presentation about our siRNA mediated gene silencing offer. And we will then follow up with a Q&A session where Cedric will be able to answer your questions. So don't hesitate to ask anything during the webinar. So yeah, I will start the presentation now.
Aussie Biosciences is a French biotech company founded 20 years ago, specialized in molecular delivery systems present in France and in the US. The French site is located in Marseille and comprises the headquarters and the production site, divided in two laboratories over 500 square meters. It's responsible for R&D and production, mRNA and LNP platforms, marketing, and sales. The US site is installed in San Diego and deals with marketing, sales, and business development. Over the years, we have settled a worldwide distributors network. One of our objectives is to develop the spread of our reagents and expertise. This expertise is based on more than 20 years of experience and innovation. Several important milestones have marked the company's history, such as the commercialization of the first magnetofection reagents in 2004, the creation of OZB-INC in 2013, and our recent acquisition of new facilities for the development of LNP and mRNA platforms. We are proud to cover a huge variety of fields, from bioproduction to viral applications, going through in vivo protein delivery or stem cell applications. All the products developed and sold, as well as our platforms, are under the ISO 9001 certification. These innovative reagents allow Aussie Biosciences to offer solutions in three main areas: cell and gene therapy, bioproduction, and custom platform productions. And considering cell and gene therapy and custom platforms, we are glad to present you today our solutions for RNA interference. We're going to focus more precisely on RNA interference mediated by siRNA, but our reagents also adapt to other mechanisms such as miRNA, for example. Gene silencing mediated by siRNA generally begins with the introduction of double-strand RNA that is recognized and processed by the dicer enzyme into short interfering RNA, or more commonly by the introduction of siRNA directly into the cell. siRNA are short double-stranded RNA molecules of about 20 nucleotides complementary to a target mRNA. Guide strand of siRNA is loaded onto RISC protein complex while the passenger strand is degraded. Once activated, RISC complex recruits other proteins such as Argonaute-2, and this complex targets messenger RNA, initiating degradation, preventing the protein to be expressed. Gene silencing implies that siRNA is correctly delivered into the cells after having navigated through several technical and biological challenges. Successful delivery results from removing limits in bioavailability that are due to sequestration or rapid degradation by serum proteins, endonucleases, and RNAs, or by the repulsive charges of the cellular membrane. This can be bypassed using transfection methods that also present some limits, such as low efficiency or toxicity. Finally, once in the endosome, siRNA should be released in the cytoplasm. And thus, in order to increase siRNA delivery, several solutions exist, but even if they each present some interest, they are still not optimal to silence gene expression. Electroporation or nucleofection are very efficient transfection methods that work well with small nucleic acid sequences, but they induce high toxicity and are less adequate to high throughput screening. Moreover, their cost is very expensive. Viral infection is maybe the most effective way to genetically modify cells, and it allows long-term expression of siRNA. However, it is technically challenging for the creation of the viral vectors. For example, impose hazardous handling and can be very expensive. On the other hand, physical methods such as microinjection or bioballistics show extreme precision and can be used for refractory cells with reduced toxicity. However, because it is laborious and needs technical expertise, it is dedicated to very specific applications, and results depend on the method used. Finally, lipofection and polyfection are often used because they are simple and quick transfection procedures that require low optimization. They show some reproducibility, but they present high yield of toxicity with a low to moderate efficiency. In response to users' needs, we've developed specific solutions depending on the application. Our solutions are non-toxic, versatile, and highly efficient, and take up the positive points described above, while eliminating the negative ones as far as possible. With our reagents and LNPs, we can address gene silencing in cell lines using Lullaby, in primary cells with SilenceMag, and also in vivo with the custom LNP-siRNA platform. Okay, so let's start with the Lullaby transfection reagent. Lullaby is a part of our catalog of patented lipids that are based on a common molecular architecture, a hydrophobic tail, a linker, and a head group that confers our transfection reagent efficiency and biodegradability, limiting toxicity. As we are both owners and producers, we can finely tune any part of the lipidic structure, the length of the carbon chain, the linker, or the nature of the head group by choosing specific amino acids, for example. Lullaby is our other transfection reagent, presents thus the capacity to form lipoplexes with an aqueous interior containing siRNA and a lipid bilayer that would serve as a cationic cargo. The protocol is simple. Lullaby is mixed with siRNA and incubated at room temperature for 20 minutes. Once the lipoplexes are formed, they are added to the cells and incubated under standard culture conditions for 12 to 96 hours before assay evaluation. And as we can see here, gene silencing is efficient when siRNA against GFP at 25 nanomolar is delivered with Lullaby in HeLa cells expressing GFP. This efficiency is demonstrated on many cell types, from cancer to fibroblast cells expressing GFP. Lullaby was used to deliver various doses of siRNA from 10 to 50 nanomoles, and the percentage of fluorescence inhibition was measured in these cell lines by flow cytometry or monitored by microscope 3 days after transfection. The results clearly demonstrate that Lullaby succeeds in silencing gene expression of at least 75% using as low as 10 nanomoles of siRNA in every cell tested. And this is clearly observed by fluorescence microscopy, where the GFP signal is almost abolished when siRNA GFP are delivered in comparison with scramble sRNA. We next compared Lullaby to other transfection reagents specific to RNA interference. In this experiment, NIH3T3 and RPE cells expressing GFP were transfected with siRNA targeting GFP with various transfection reagents according to the manufacturer's protocols. After 72 hours, fluorescence intensity was evaluated by flow cytometry, and percentage of GFP inhibition was calculated against the non-treated cells. In these specific cell types, Lullaby induced the highest gene silencing and outperformed competitors with more than 75% of fluorescence inhibition. Finally, toxicity was also evaluated since it is the other main issue when delivering siRNA with transfection reagents. C2C12 and NIH3T3 expressing GFP were transfected with 25 nanomoles and 10 nanomoles respectively of siRNA GFP with various transfection reagents. After 72 hours of incubation, both gene silencing, represented by the bars, and viability as the red dot, were monitored. In these cell types, the results demonstrated that either Lullaby performed as well as the most efficient transfection reagent, but it is far less toxic, or for the same viability than competitor, Lullaby performs better. Now let's have a look at the scientific publications. I'll start with the short and non-exhaustive literature review by highlighting the study done by Emma Shanks of the Cancer Research at UK Beatson Institute in 2014. The goal of this work was to develop a strategic siRNA screening approach in cancer cells. And for this, a library of 26 transfection reagents was collected and tested for gene silencing and cell survival on more than 20 cell lines. siRNAs were screened through several thousand experiments, and some reagents were efficient but toxic. The overall conclusion of Professor Shanks was that by far their preferred reagent is Lullaby from OZ Biosciences. They have used this reagent in over 20 cell lines and have found it essential in enabling siRNA screens in hard-to-transfect cell lines with minimal toxicity. This is one of the reference articles about efficiency and the low toxicity index of Lullaby. In this next example, Lullaby was used to deliver 50 nanomoles of siRNA targeting key proteins for metastasis first in cancer cell lines cultivated in 2D and in 3D multicellular spheroids. Western blot analysis confirmed that siRNA specifically knocked down target gene expression. The number of TKS5 positive invatosome and type I collagen cleavage demonstrated the efficiency of Lullaby-mediated gene silencing in 2D. Moreover, in 3D multicellular spheroid embedded in collagen, Lullaby succeeded in silencing gene expression of key genes as observed with actin staining. In parallel, 3D invasion measurement demonstrated that proteins implicated in caveoli are required for 3D invasion of breast cancer cells. Thanks to the versatility of Lullaby for 2D and 3D models, the authors showed that invasion by Cav1 depleted spheroids over a 48-hour period was decreased by 40%. In this work by Whitlaw, Lullaby was used to silence gene expression in B16F1 cells using a pool of siRNA targeted ERC1 to modulate adhesion. The hypothesis would be that ERC1 depletion would mimic CYRIB loss on focal adhesion. Western blot, as well as experiments on ERC1 expression, cell area, and focal adhesion measurements confirmed the capacity of Lullaby to efficiently silence gene expression. As mentioned by the authors, using a pool of small interfering RNAs, siRNA specific to ERC1, they achieved a greater than 70% reduction in ERC1 protein levels. Finally, functional experiments showed that B16F1 with depleted ERC1 displayed a larger area as observed in CYRIB knockout cells. And considering primary cells, we've developed SilenceMag, a magnetofection-based transfection reagent for RNA interference experiments. SilenceMag is based on our proprietary and patented technology, magnetofection. The central point of this transfection method is the magnetic nanoparticles that are formed by an iron core and a pre-coating layer, allowing to engraft the various coatings of different materials and charges. The protocol is very simple. Once the complexes of siRNA and SilenceMag are formed, they are added to the cells, and the plate is then incubated for 20 minutes on a magnetic plate. And we can see here the efficiency of GFP silencing in C2C12 cells using this technique. Now let's take a closer look to what happens in detail. Once the magnetic complexes are formed, they are added to the cell culture medium. The cells are then placed onto a magnetic plate for 20 minutes of incubation. At this time, the specific magnetic field represented here by the B vector attracts the magnetic nanoparticles onto the cell surface within minutes. Under the influence of this physical force, the magnetic nanobeads are clustered and enter the cell through classic endocytic pathways such as clathrin or cavalin pathways. Importantly, no hole is created, leaving the cell membrane intact as opposed to other physical methods such as electroporation. Inside the cells, the complexes are then located into endosomes from where they will evade due to the action of proton sponge effect. The beads will be sequestered in vacuoles, excreted out, or be degraded by the cell's iron metabolism, while siRNA will be released and dock to RISC complex to target mRNA of interest. This unique technique allows to bypass the classic transfection limits, as most of the dose comes into contact with cell surface very rapidly. Magnetofection overcomes the poor adhesion occurring, depending on cell types, and favors endocytosis of the complexes triggered by the clusterization of the beads on the cell membrane. SilenceMag is really efficient to silence gene expression, especially for hard-to-transfect cell lines or primary cells. And this is what is illustrated here. We can see that GFP expression is silenced after transfection with siRNA against GFP compared to scramble siRNA. SilenceMag allowed to induce also gene knockout in a dose-dependent manner in cell lines expressing LAC-Z. And finally, SilenceMag is really efficient for sensitive and hard to transfect primary cells, such as HUVEC, and demonstrated here where TAL1 transcription factor expression was silenced after transfection with magnetofection. If we take a look at the literature, in this first example, the authors transfected 100 nanomoles of siRNA targeting human cavalin 1 and cavin 1 PTRF in HUVEC. The goal was to deplete specific components of cavalae and investigate their role in the formation and dynamics of transendothelial cell macroaperture. We can observe that following siRNA treatment in HUVEC, CAV1, and PTRF depletion induced tunnel creation that we can visualize by actin staining. Moreover, TEMs in cells where cavalin 1 was depleted were wider with a 5.4-fold increase compared to controlled cells, and their area was also dramatically improved. As the authors mentioned, the CAV1 and PTRF was reduced of approximately 80%, allowing them to study how these proteins impact TEM formation and dynamics. In the following example, primary canine nose keratinocytes were transfected using 50 nanomoles siRNA. The goal of the experiment was to mimic the loss of function phenotype observed in KNPK affected dogs and confirm that the SUV39H2 variant was the functional cause of the HNPK phenotype. Keratinocytes were transfected with combination of siRNA. We can see here that the combination of siRNA 2 and 3 achieved dramatic gene silencing. That's verified by the immunostaining. The authors confirmed the efficient SUV39H2 knockdown at day 0 and 3 after calcium switch by RT-PCR and immunoblot that allowed them to demonstrate the implication of this gene in HNPK phenotype. This last example of SilenceMag is very interesting since it will allow us to bridge with the next section. So on the basis of SilenceMag, we developed an in vivo version that was used to silence gene expression of KIR 4.1 after intrathecal injection into the subarachnoid space of neonatal mice. The goal was to disrupt KIR 4.1 channel in the spinal cord by targeting magnetic nanoparticles thanks to an external magnetic field. Fluorescence microscopy confirmed that the in vivo silencing is effective since we can observe a loss of KIR 4.1 staining. The gene knockout was then confirmed by RT-PCR and further showed a direct impact on membrane potential and voltage clamp. As the authors stated, a decrease of more than 51% in KIR 4.1 mRNA was measured in the spinal cord after injection, allowing them to reveal the impact of this gene on astrocyte excitability and neuronal discharge pattern. Even if Lullaby and SilenceMag are very efficient and non-toxic, there's still some room for improvement. You can optimize conditions of transfection by playing with siRNA dose and transfection reagent ratio. Or you can perform a reverse transfection by adding first the complexes and then the cells. It is also possible to perform sequential transfection 24 hours apart from the first one. For this, we recommend slightly reducing the doses and volumes. And you can use any medium or cell culture system. Finally, a unique feature of magnetofection is that you can change right after magnetofection procedure for sensitive cells. The magnetic plate keeps the complexes onto the cell surface while the medium is carefully removed. If we have a look at the publications related to gene silencing, we can see that within the lipid-based transfection reagents, Lullaby is the most described in literature, with more than 70% of the articles. And concerning the magnetofection, SilenceMag is also the most cited. The difference here is less visible since we offer various cell types and transfection reagents that are also used for gene silencing, such as NeuroMag or GlioMag or XPMag dedicated to XPlant. As a matter of fact, the repartition of the science articles is almost 50-50 between magnetofection and lipid-based, with the predominance of the cell lines for lipids. Specifically, SilenceMag was preferentially described in primary cells and Lullaby mostly for gene silencing in cell lines. As we can see here, both reagents can deliver any kind of short nucleic acids, but more than 80% the publications mention RNA interference using siRNA. Finally, with both reagents, we can address cell types from any territories, from canine to fish or tick cells even, as long as the majority of the silenced cells are from human origin. In addition to in vivo SilenceMag, our LNP platform has developed expertise in encapsulating siRNA more designed to in vivo applications. LNP-siRNA have a great potential not only in research, but also show great promises in therapeutics, since many LNP-siRNA-based drugs are approved by the FDA, such as, for example, Onpattro, for treating transthyretin amyloidosis. This is due to the characteristics of these vectors to efficiently deliver the siRNA in various sites of the injection depending on LNP generation. Moreover, these vectors are very stable, conferring a prolonged effect, usually up to 6 months. Chemically speaking, the versatility and the adaptiveness of the LNP mainly comes from their composition, with the choice of the ionizable lipid or their decoration with targeting ligands. The creation of any kind of LNP with our own ionizable lipid or existing ones can be addressed by our LNP platform. The particularity of our LNP platform resides in the fact that we are the owner and producer of our own ionizable lipids. This means that we can play with the size, the shape, the properties of the lipid nanoparticles. The ionizable lipids, in association with helper lipids, cholesterol and PEG lipids favor the formulation of monodisperse LNP with narrow size distribution and high encapsulation efficiency, as illustrated here by TEM or DLS with RNA-loaded LNP. All our LNP are extensively qualified, and a superior grade quality control is also available on demand, including mycoplasma and endotoxin detection. The formulation is done by microfluidic or jet mixer, where the active ingredient is mixed with lipids before adjustment by dilution. The system of mixing depends on the application and the volume required. Our ionizable lipids were extensively evaluated in vitro. First, we used HEK cell line transfected with GFP to screen 10th of LNPs based on our lipids. We chose the lipids that induce the most important gene silencing, as illustrated here with SS18 and SS23. Then the best candidates were validated in a more functional model to deliver siRNA IL-1 and PMA-activated THP1 primed with LPS. As an example here, the dosage of IL-1 by ELISA demonstrated the effective silencing induced by siRNA IL-1 LNP formulated with SS18 and SS23. The results were as good or even better than with knockout by Lullaby. These lipids are currently evaluated and validated in vivo by partners in arthritis model and rabbits as part of the SimPain European grant. Today, our platform enables efficient encapsulation of any nucleic acids from DNA to RNA, including siRNA, with concentrations ranging from 0.1 to 1 milligram. Particles as well as the RNA inside can be fluorescently labeled for an effective monitoring in vitro and in vivo. We can play with the size and the charge of the particles and also add targeted ligand on the surface such as sugars, peptides, or nanobodies for tissue-specific delivery. And we offer flexibility in lipid composition using our proprietary or commercially available ionizable lipids. This versatility makes our platform ideal for both research and therapeutic development. So what I'd like to emphasize before ending this webinar is the fact that for more than 15 years now, Lullaby and SilenceMag have been cited in hundreds of high-ranked journals. This success can be explained by the fact that these dedicated siRNA transfection reagents are non-toxic and allow efficient gene elimination in cell lines as well as in primary cells. Even if most of the results were shown with siRNA, Lullaby and SilenceMag are highly efficient for any short nucleic acid sequences, such as miRNA. All of these features render them ideal for high-content screening.
So, thank you for listening to our presentation. You can contact us at contact@aussiebiosciences.com or for any technical question at tech@aussiebiosciences.com. We are now going to answer some of the questions that were asked. So the first one is, you talked about short RNAs, but what about guide RNAs? Can we use your reagent for genome edition with CRISPR-Cas9?
Yeah, guide RNA are so small RNA, so they are totally compatible with either Lullaby or SilenceMag, and we can also encapsulate them in our LNP. Now, if we talk about genome edition with RNP complexes, this is a totally different application since we have to deliver a protein, for example, Cas9 protein associated with a short guide RNA, and this form a ribonucleoprotein complex. This is big. And for this, we have developed a specific transfection reagent, and I will just recommend to choose Razor Pro delivering CRISPR that was designed to transport both the protein and the short guide RNA. And of course, for any other short RNA, such as miRNA, as we said during this webinar, we can use Lullaby and SilenceMag. It will be, of course, working.
Thank you. Another question is, can we transfect tissue explant with your reagent? So they are working on retinal explant, and they ask if SilenceMag would be adapted.
Yeah, that's a very good question. And yes, SilenceMag could be adapted. The point is that transfection of organotypic slice or explant is really tricky, and magnetofection could be is one asset to transfect them. And speaking about specifically retinal explant, during a European project on ocular disease, we have developed a magnetofection reagent that we called XPMag, and that was specifically developed to deliver a siRNA in retinal explant. And to make it works, we have changed the direction of the magnetic field, and now the magnetic plate is positioned above the cell culture plate, above the explant, so that the complexes that are added into the medium are then attracted upward the layer of the retinal explant. And it worked well. We have many publications on this, and I can cite the work by Marco Bassetto, who was a student here and who developed this technique during his PhD thesis. So XPMag would rather be my recommendation for silencing in retinal explant.
Thank you. Another question is, are the reagents compatible with multiple transfection? Is it possible to repeat the silencing?
Yeah, of course. Both reagents are totally non-toxic, and so they can be used sequentially. And as we talk in the webinar, for sensitive cells, the best recommendation would be to lower a little the efficient optimal dose so that the cells do not get saturated by the complexes. Otherwise, there is no other precautionary measure to take. And yeah, in the webinar, we talk also about the work by Montero et al., who transfected the cerid in 3D, and we used sequential transfection 24 hours after the first one to silence gene expression in 3D. So yeah, definitely we can perform sequential transfection.
Thank you. Now we have some questions regarding LNPs. So do you know what are the endogenous receptors for taking up the LNP? And also, can we direct the vector through desired territory?
Yeah, up to now, the specificity of the receptor of the LNPs are still under investigation. It is known that some LNPs can interact with cell surface receptors, such as lipoprotein or scavenger receptor. But the point is that LNPs are mainly delivered into the liver, and one receptor of the liver is apolipoprotein E. But if we consider naked LNP, the delivery of the cargo highly depends on the composition of the LNP, its composition depending on the lipids, the colipids, the concentration of PEG, and of course, depends on the charge and the size of the LNP. And in the lab, we have tested our iodizable lipid issued from our library in vivo, and we have observed that depending on the nature of the lipids, the LNP would preferentially go to one territory or another territory. So even if we don't use receptor, we can still target the territory, and our platform has a capacity to functionalize or decorate the LNP with specific marker to target some territories.
Thank you. And the question, what is the size of the SilenceMag magnetic beads?
The size vary between 150 and 180 nanometers.
Okay, so this will be our last question for today. I see there are some questions that was left unanswered, but we are going to answer them by email, so everybody will have a response. If you could stay until the end of this webinar, we have a quick survey to ask how you found this webinar, and it will be really helpful for us to have a feedback. Thank you very much, everyone, for attending this webinar, and have a nice day.
Bye.
Bye.